So yeah, as you mentioned, I just recently joined as a permanent member of academic staff within the School of Biology and Environmental Sciences, but I've actually joined the school about a year and a half ago, more on a temporary basis. And so for those of you who have seen my job interview about a month ago for the permanent position, I have to say there's a little bit of overlap in this presentation today, but I have added in a little bit of additional information towards the end, so I think it's going to be worth it to stick around. And yeah, so as Taz mentioned, I'm interested in wetlands specifically, but in general, I'm really interested in ecosystem trace gas exchange. So I usually work at that interface between biosphere and the atmosphere, and looking at exchange such as water, carbon between those two, but also things like energy or other greenhouse gas fluxes. I've worked with in the past, and I've worked across a number of different ecosystems in the past as well. So I have worked in urban systems as well and agricultural systems. But today I'm going to focus on my work in wetland systems, and I'm going to specifically focus on water exchange. I know I mentioned carbon in my title, but I'm not going to talk too much about that today. Just to keep things under 10 or under 15 minutes, I guess, just focusing on that. And that's kind of the work that I've been doing over the past year and a half, and that I'm really wanting to focus on in the near future. All right, so to get started, I just wanted to give everybody a little bit of background, because I know we have a lot of diverse interests in the Earth Institute. So when I talk about ecosystem trace gas exchange, so if you think about a dry land system, such as an agricultural field, for example, we have the couple of main fluxes that we think about in this context. So CO2 uptake, for example, through photosynthesis, but then also release of CO2 through plant or soil respiration. And then we have water flux, which is the other main flux. So one that's the most important part usually in dry agricultural systems is transpiration. That comes through the stomata and is at that level also linked directly to the carbon flux. So there's this carbon-water trade-off usually associated with these systems, where carbon uptake through photosynthesis also is associated with water loss through transpiration. And so that transpiration flux is a quite important component in terms of water loss for these systems. We also have evaporative flux of water, so that's evaporation from the soil or from other surfaces directly. And in dry systems, that usually takes on a fairly minor role. But if we switch then to wetland systems, some of these fluxes change quite significantly. So for example, in the carbon area, soil respiration does not play a major role in flooded systems anymore. But we do have to consider another carbon flux, and that is methane, that is often quite important in these flooded systems, and not just as a form of carbon exchange, but also because methane is a very potent greenhouse gas, of course. So it does play a really, very major role for wetlands often in terms of their greenhouse gas budget. As I said, I'm not going to talk in detail about carbon today. So looking at the water fluxes, we can also imagine that evaporation component might change quite significantly when we move from a dry system to something that has water available at the surface at all times. And so that's kind of what I want to look at in a little bit more detail today, and show some of the research that I've done in that area. And just to remind everybody why we care about these water fluxes to begin with, just to show the terrestrial carbon water cycle here. So that transpiration part from plants is actually a very big component within the water cycle of adding water vapor to the atmosphere that then gets turned into precipitation and returned to the land in this water cycle. So it is a very big component in the water cycle, not just in the global water cycle generally speaking, but also specifically in certain ecosystems that can be a very major component. So if we think about, for example, the Amazon or other rainforests, that transpiration really plays a major part of driving that water cycle in these systems. And so it is important that we get a good handle on how much transpiration is added to the atmosphere from these different ecosystems. And that's also specifically important if we think about climate change and how climate might affect the water cycle in the future. We can see that transpiration from plants, because it is linked to the carbon cycle through the stomata, and because it is linked to that biological component of plants, it might respond quite differently to things like temperature change or a rise in CO2 levels, for example, compared to an evaporative component that is more kind of a physical component and not linked to that biological part. So being able to understand how transpiration changes as environmental conditions change, such as temperature or CO2 levels, that is also a really important thing to know, so that we can hopefully predict how the water cycle might respond to future climate scenarios. And so I had the opportunity to study the water cycle, and specifically evaporation and transpiration, more detail in these different wetland sites that are located in the Sacramento-San Joaquin River Delta in California. And California, water is a very important topic in California specifically because it is famously drought-prone climate there, and they do have a lot of water shortages. So the water cycle is a very important topic. And so I was able to study the water cycling of these different wetlands. And they provided a really unique opportunity because they're all very close in proximity. So they experience the same climate. They're all within a few kilometers of each other. But we had quite a gradient of the structures that these wetlands represented. So on the one end, we had a wetland like Mayberry, where you can see from this picture here, we had quite a lot of these open water areas and channels and pools that provided large open water surfaces interspersed with these denser patches of vegetation in that wetland. And then on the other side, on the other extreme, we had West Pond wetland, which is a very densely vegetated wetland. And so this wetland does not have any open water channels or pools at all in its footprint. And it's very densely packed vegetation. And then we had some that were kind of in the middle, in between. So East End here is an example where we had a little bit of open water, but we did have a lot of dense vegetation as well. And just to remind you that even though it looks like a very dense mat of vegetation here in West Pond, almost kind of like a meadow or something, it is a flooded system. So there is about a meter of open or of water at the bottom of this vegetation. So if you do walk through this, you will get wet feet. I can guarantee you that. So it is still a very water, there's still a lot of water available in the system, even though it doesn't look like it in this picture here. And so the methods I used to study the water exchange in these different systems. So on the one hand, I was looking at some leaf level measurements. So using these chamber methods, where we can then look at the transpiration component directly for these different leaves. And we are also able to control the environment within that chamber. So we're able to control, for example, the dryness of the air or the temperature or the CO2 level within that chamber directly. And then we can see and watch how these different leaves respond to those changes in environmental conditions. And we're also able to look at the different plant species that are present in these wetlands. So in these wetlands specifically, we had two, the main dominant species were two different species. So there's a native tule species, which is seen, which you can see in this picture here. There are these kind of brown rod-like leaves. And then there's also the invasive cattail. And depending on the wetland, they some are more dominated by one versus the other, but in all wetlands, both of these appear, and they're kind of both of importance in these wetlands. And so with these methods, we're able to look at these two species separately and see if they have any differences in how they respond. But then I was also interested in the ecosystem level evaporation and transpiration as well, because it can be very difficult to scale up these leaf level measurements to the ecosystem level, especially in these wetlands that can be very heterogeneous, not just in terms of their spatial contribution of open water pools and dense vegetation, but also, as I said, the species composition can be quite different. And so it can be quite difficult to scale these leaf level measurements up to a meaningful ecosystem level response. And so we used a method called eddy covariance to get ecosystem level evapotranspiration measurements. So that's the total water loss from the system. And then I was pioneering and developing this tool where we use machine learning to then separate out the evaporation and the transpiration component for these wetlands. And so showing a little bit of data now. So this is the leaf level measurements, just as an example. In the top panel, we have the carbon exchange. So the photosynthetic carbon uptake shown here for the two different species. And then in the bottom panel is the water exchange. So that's the water loss from the same leaves. And so this shows a diurnal cycle. So starting at midnight, going to midnight. On a bright sunny day at the peak of the growing season. I believe that was in early August or late July where we took those measurements. So you can see kind of like the general response of photosynthetic carbon uptake during the day and not much happening in terms of carbon exchange at night. And then again, focusing on the water exchange here, one thing that we were able to see here is that there was a little bit of a difference between the two species actually. So if we look at the dashed black line here, that's the vapor pressure deficit. So that's a measure of the dryness of the air. And we can see that in the early afternoon here, we had a very high VPD. In this location, so this being California, that's not something that's unusual to happen in the summer. We get these very dry, very hot days very regularly there. And we can see that the native tule actually tracks the water exchange, the transpiration, tracks that vapor pressure deficit curve quite well. Whereas for the cattail, that's not quite the case. And so it looks like the cattail did restrict some put some restrictions on the stomata and restrict the transpiration, especially in those very hot and dry afternoon hours. And that had an impact on the carbon exchange as well. So that was quite interesting to see how these two different species responded differently to the different climate conditions and different environmental conditions. And then looking at the ecosystem level, so here I'm showing annual data across a number of years. And so if you focus on the top left panel here first, that's the total water loss at the ecosystem scale for two of these wetlands. So the blue is Mayberry, which is the one that had a lot of open water areas. And then the red is West Pond, which is the one that was the very densely vegetated wetland. So these are the two extremes, basically. And what we can see is that the annual water exchange across these years between those two wetlands isn't that different. We do see that in the first few years, Mayberry tends to have a little bit of a higher water exchange, but overall, on average, they're very similar. And then looking at the carbon exchange, so that's the photosynthetic carbon uptake for these two systems for each year. Again, there are some years where we see a little bit of a larger difference, but overall, on average, they are very comparable. So on the ecosystem level, they don't look too different in terms of their water and carbon exchange. But if we then go into the individual components of evaporation and transpiration here, we can see that Mayberry has a much higher component of evaporation compared to West Pond, and the opposite is true for transpiration. So if we think about how much transpiration contributes to the total water loss, we can say that West Pond is very much a transpiration-dominated water wetland, whereas for Mayberry, it's more of an evaporation-dominated system where the two, transpiration and evaporation, almost contribute the same amount. with these types of information now. And yeah, so just to kind of sum this up a little bit, so we had this gradient from more open water to more closed vegetation in the wetland, and we were able to observe some of these differences. The one that I focused on was that the closed vegetation wetland was more transpiration dominated and the open water was more evaporation dominated, but we actually were able to show some differences, for example, in water temperature as well. And that water temperature also drove differences in evaporation as well. So we did see some really interesting transitions and changes as we moved through these gradients for these different systems with the water exchange. And just a very brief note at the end. So I'm currently working on developing some projects where I look at some of these methodologies a little bit more in detail, looking at the partitioning of transpiration and evaporation for the ecosystem level. But also I'm really interested in looking at this in an Irish context specifically. And I think that within Ireland, it's really interesting to look at that, not just within wetlands and flooded systems, but generally because Ireland usually has a very wet climate, not this spring, but usually we do see a lot of rain and precipitation events, which can lead to interception of the water on leaf surfaces, and therefore could have a very high evaporative component, even for these dry systems, such as agricultural systems or forest systems specifically. I think would be very interesting to study what the water exchange components look like in these Irish systems specifically. And with that, just acknowledging a couple of collaborators, students, technical staff, and funding agencies, without all of whom a lot of this would not have been possible. And I thank you for your attention, and happy to answer any questions and look forward to a discussion about these ideas now.